everyone. Thank you very much for joining us today, and thank you, LabRoots, for the invitation. So, my name is Hailey, and I am a scientist working at CN Bioinnovations. I'm really excited to be here today to talk about how primary cells could be incorporated into a gut-liver MPS models for application in ADME studies. So here is an overview on today's talk. To start off, I will be giving a brief introduction into what ADME is and how is it related to the preclinical drug discovery system. I will then explain the concept of microphysiological systems, or MPS, and how we use this technology to address the current challenges of the preclinical tools. Finally, I will be presenting a few case studies on how MPS models could be more predictive of ADME profiles of drugs compared to the standard preclinical tools. So, what exactly is ADME? ADME is the abbreviation for absorption, distribution, metabolism, and excretion, which are the four key processes to describe what happens to the drug following administration into the body. Because it describes the behavior of a drug, it is therefore a crucial measurement in preclinical studies to investigate various drug properties. So, drug properties can include bioavailability of the drug, and also their safety and toxicology profiles. So while the market is constantly in a huge demand for new drugs, the whole drug discovery process is unfortunately very lengthy, very expensive, and has a low probability of success. So out of the 10 to 15 years in the whole drug discovery process, there's around 6 years is spent in the preclinical drug discovery process, and that accounts for around 33% of the total cost before even releasing to the clinical trial phase. So even if the clinical trial phase is reached, it still takes around 8 to 9 years for a drug to be completely released into the market if successful at all. But unfortunately, only 10% of the total drug developed can actually be released into the market, and for various reasons. Reasons for drug failures include a poor drug efficacy, followed by some safety issues that has not been picked up in the preclinical testing. Other factors can also include poor drug-like properties and also market assessments. To estimate the ADME in preclinical study, scientists have a wide range of tools available to them for evaluating new therapeutics and molecules. So one of the most common approach is the in vivo approach using animal models, which I'll go through the benefits and the challenges in the next slide. Some scientists will also be using 2D cell culture and 3D organoids for their studies, and this is part of the in vitro toolbox. So in vitro methods sometimes could be used in conjunction with in silico methods such as mathematical modeling to estimate the ADME profile of a drug. So let's start off by talking about the benefits of the models. So the reason why animal models is still a common approach for drug studies is because of the ability to model the whole drug body interaction. So some animals, such as mice, are easily manipulated to accommodate different dosing conditions. That could include different dosing regimens, and they also have the ability to model both acute and chronic dosing. However, some challenges associated with the use of animal models include the limited physiological relevance to human, and it is also relatively lower throughput. It is because in most cases, multiple animals will be required for one single study, which also gives rise to issues such as an increase in research costs and also ethical concerns. Some of the challenges associated with animal models can be addressed by in vitro models. Most of the cell lines are relatively easier and cheaper to manipulate, which could offer a more cost-effective and high-throughput solution compared to animal models. However, some limitations include the short-term culture, and it has also been really challenging to culture a multi-organ model of using in vitro approach. And one of the most significant limitation with regards to in vitro model is that most cell line has a very low expression of human relevant transporters and enzymes, which makes it particularly challenging if we want to use an in vitro approach to measure the ADME profile of a drug. So, unfortunately, all these limitations have significantly slowed down the progress in drug development. So, over the recent years, there has been an increasing number of new chemical modalities being proposed, which needs to go through the long and complicated process of drug discovery. So, in addition to the challenges I have just mentioned, there has been further threats to the timeline and cost regarding the use of animal studies. So, one additional challenge is the limited access to non human primates for these preclinical studies, which has directly acted as a barrier to slow down drug development. So, another challenge is the pressure from the global regulatory landscape, which I'll go through briefly in the next slide. So, as I've previously mentioned, our research industry is currently facing an increasing ethical pressure to reduce the numbers of animal used. So, one example is the FDA Modernization Act that was approved in the United States back in December 2022. The Act has removed the mandate for animal study at preclinical stage. Instead, it has proposed that MPS and organ-on-a-chip are acceptable alternative techniques that can be used to demonstrate a drug's efficacy and safety in the preclinical state. So, other examples include introduction of new rules all over the world or having a common goal to reduce the use of animals testing in the preclinical stage. So, in order to address the limitations of the current preclinical toolbox and also to address the pressure from different regulatory bodies worldwide, there has been an increased effort to develop new approach methodologies as a better alternative to animal studies. So, in particular, there has been an increasing evidence suggesting the use of microphysiological systems, and they can generate more reliable and human relevant preclinical data. This could be used in combination with the traditional preclinical models in order to further bridge the gap between preclinical and clinical studies. So, for those of you who are not familiar with what MPS is and what does it do, here is a brief overview on the technology. So, MPS are also known as organ on a chip, which are designed to better resemble human biology in vitro, and they are characterized by a few common features. So, one key feature is the use of human cells and tissues in MPS. So, the human cells in MPS are typically grown in three dimension and with geometrical confinement or patterning. Another characteristic is the presence of fluidic flow. So, fluidic flow within MPS is designed to mimic blood flow in the human body and can provide oxygen and nutrient within the culture. Other key features include the ability of MPS to produce a tightly controlled environment, and some of them are even equipped with a specific sensors to closely monitor culture conditions. So, how exactly do MPS work? MPS technology is usually a combination of hardwares, consumables, and assay protocols that enable a user to recreate the complex biological structure of human organs. So, one example is the liver model. A liver model features a perfusion of media within each well, which in turn allow formation of 3D microtissues. Another example is the barrier model, which features a basolateral flow within the plate. So applications include a lung or gut model, and a barrier model could be used to study the transport of substances across a membrane. Finally, there is an option of dual organ model in MPS, which links a barrier model with a liver model. So within a dual organ model, there is interconnected flow between the two organs, which allows a user to simulate inter-organ crosstalk for a wide range of application, which I'll go through later on in the presentation. So while each of the MPS organ model have their specific applications, these three are the key applications that are all linked by the liver. So today we will be focusing on demonstrating how these organ models are predictive of human biology and how this could be incorporated into your normal research workflow to improve your research efficiency. So, in the following slides, I will be expanding further on how we model the human liver using our MPX technology and how this is able to resemble the structure and functionality of an actual human liver. So, to build a human liver model in vitro, there are a couple of crucial hepatic features that must be recapitulated. So, a functional human liver is characterized by a number of activities that includes albumin production and urea synthesis, which were produced by hepatocytes within the liver. So, a functional human liver is also characterized by different markers of hepatic damage, which includes ALT and AST release, LDH production, and also the release of cytokines. Other important factors to consider include the expression of various metabolic enzymes and transporters. It is because liver is a key site for metabolism of many drug compounds, and therefore it is very important that an in vitro model is able to reproduce these features, especially when used in applications such as ADME studies. So, to build a human model of liver, first we need to seed validated cells on a collagen coded scaffold within the well. And after cells have been seeded, there will be fluidic flow within the MPS system to allow perfusion of media. This perfusion of media ensures a constant supply of oxygen and nutrients that allows formation of a 3D microtissue structure. So the liver model has been successfully characterized both functionally and structurally. So this panel of microscopy images demonstrates the development of an MPS liver model over time, and it could be demonstrated that a 3D microtissue structure was formed within 7 days. So after a 3D microtissue has been formed, the model was also stained positive for the expression of biokinetically, which is a key hepatic structure. This culture was also positive for the expression of some key structural biomarkers, including a cytoskeleton and tight junction proteins. So, when analyzing the MPS model for the functionality, the MPS model demonstrate a higher metabolic activity and also higher rates of albumin production than the traditional spheroids and sandwich culture. This indicates that the liver model generated using the MPS technology can demonstrate an improved functionality for downstream applications. So, when estimating the ADME profiles of a drug, it would be particularly useful to have a gut model that accurately reflects the scenario in the human intestine. However, the current standard for gut model are the use of immortalized cell line, which I've previously mentioned. They are usually characterized by a very low expression in transporter and metabolic enzymes. So, in the next few slides, I will be explaining the potential of using a primary gut model in accurately reflecting the barrier function of human intestine. So, to generate a primary gut model, we collaborated with Altus Bowel System and established a gut barrier using a replica platform. So, the gut barrier was derived from a crypt epithelium stem cells that were isolated from human genome samples. These cells were then expanded on a biomimetic scaffold in static condition. So, here is a diagram representing the setup of the primary gut model. The culture then go through two major phases, which are the expansion and differentiation phases. Each are characterized by a specific pattern of gene expression. So a fully differentiated barrier should be ready in around 2 weeks' time, and they have been shown to express key intestinal markers. So we have also characterized the primary gut model and that they demonstrate three key features that show they could offer a more human relevant option than the standard KCO2 model. So the first feature is that the primary gut model demonstrates a distinct and continuous layer of mucus, which closely resembles the inner layer of the human intestine. The second feature is that the primary gut model has a distinct proliferative and differentiated population. Plus, as I've just mentioned, they were ready in just 2 weeks' time. So, compared to the standard KQ2 model, which takes around 3 weeks to complete the differentiation process, a primary gut model could be a more efficient approach. Uh, Further qPCR analysis also suggested that primary gut model demonstrate a higher expression of key ADME biomarkers that includes the expression of a transporter and metabolic enzymes. So this would therefore be more suitable for downstream ADME studies. So, considering that both liver and primary gut models have been characterized to demonstrate a higher human relevancy compared to traditional cell lines. So, in the following slide, I will introduce the concept of using MPS to combine these two primary models and how this could be used to accurately model the gut liver axis. So, what's so attractive about a multi organ MPS? What's so attractive about this technology is that a multi-organ MPS features a fluidic flow which links two separate organ models into one single plate, which could in turn allow the modeling of a complex disease process, biodistribution of molecules, and also inter-organ crosstalk. So in this case, we are modeling the gut-liver axis, and this could be represented by a diagram in the bottom left. So a gut model could fit into the transwell compartment within the multi-organ plate, which is in turn linked with the liver culture. So by modeling the gut-liver axis, it introduces the flexibility of the model to simulate both organ or intravenous dosing in human, which I'll go through in more detail later. With the help of the fluidic flow, they also introduce a potential for an improved PK and efficacy prediction, and also has the option to be used in conjunction with mathematical modeling for an improved translation to human. So once the gut model has been fully differentiated, it could then be transferred to the transwell compartment within the multi-organ plate and establish the gut liver co-culture. So we have also established a special co-culture media that retains the functionality of the model for at least 48 hours. So at the end of 48 hours, we performed some measurements and demonstrated that a primary gut barrier remained intact following exposure to a fluidic flow and also exposure to the liver culture. And in terms of the liver culture, the functionality of the liver model in co-culture is comparable to the single organ model control. And this could be reflected in terms of various factors such as the albumin production and CYP3A4 metabolic activity. Finally, to validate the collective metabolic capacity of the gut and liver models, we dosed the model with a compound called 7-HC. Results suggested that the MPS model has been able to metabolize the compound, and this could be done so either by just the gut or liver model alone, or collectively as an interconnected gut liver model. So, the ability for this gut liver MPS model to metabolize 7-HC indicates that the model might be suitable for ADME studies for more drug compounds. So, as I've previously mentioned, the interconnected gut liver model has the flexibility to simulate both oral and intravenous dosing of different drug compounds. So, for oral dosing, the drug is added directly onto the primary gut barrier at the apical compartment of the transwell. So, depending on the transport mechanisms of the specific drug, the drug can be transported across the gut barrier into the circulating media underneath and eventually come into contact with the liver culture. The multiple fluidic flow in the gut liver MPS also allows the modeling of the combined effect of intestinal absorption and hepatic metabolism of drug compounds. So, in terms of intravenous dosing, the drug could be added directly into the liver compartment. This setup models the scenario where a drug was injected directly into the bloodstream and eventually travels to the liver for metabolism. To estimate the ADME profile of the drug of interest, media samples could then be taken at multiple time points and analyzed by LCMS to determine the changes in drug concentration in the circulating media over time. So the first drug we tested the model with was temocapril. So temocapril is a drug that is rapidly absorbed into the bloodstream through the gastrointestinal tract, where once it's entered into the bloodstream, temocapril was then converted into its active metabolite temocaprilat by an enzyme called carboxylase 1. So, temocapro is actually designed to be resistant to intestinal hydrolysis and that the actual human intestine do not express the isoenzyme CES1. However, for a traditional KCO2 model, there is positive expression for CES1, which obviously differs from the actual scenario in human intestine. Therefore, if a researcher were to use a KCO2 model to estimate bioavailability and ADME profile of temocapro, it contains the risk of overestimating temocapro clearance in human body. So, on the contrary, primary gut cells derived from the jejunum do not express any CES1, and that it correlates with the expression pattern in the actual human intestine. So, the media samples were taken and analyzed by LCMS to measure changes in the temocapro concentration over time. So, first of all, temocapro could be metabolized by the liver only and both the gut liver models. However, when comparing with the KCO2 model, there has been a higher concentration of temocapro in the circulating media in the primary gut model over the course of 48 hours. We have also measured the relative amount of the metabolite temocaprolate in the model, and the amount present in the primary gut model was much lower than what was present in the KCO2 model. So there was also a larger proportion of parent drug temocapro remaining in the primary gut than the KCO2 model. So collectively, all this evidence has suggested that temocapro might demonstrate a resistance to intestinal hydrolysis by our primary gut liver model, which is also in line with the CES expression pattern in human intestine and also the PK properties of the drug itself. The drug that we tested the model with was midazolam. So midazolam was primarily metabolized by CYP3A4. There has also been increasing evidence suggesting that the intestinal sorry So another drug that was tested was midazolam. So midazolam was primarily metabolized by CYP3A4. There has also been increasing evidence suggesting that the intestine is an important site for first pass metabolism for this drug, in addition to the human liver. And as I've previously mentioned, our qPCR analysis suggested a higher CYP3A4 expression in the primary gut model than the KCO2 model. This again suggested that the primary cells can possibly provide a more accurate estimation on the ADME profile of this drug compared to the traditional KCO2 culture. The results of midazolam dosing suggested that there is a fractionally higher clearance in the model containing primary gut cells than the KCO2 cell line. Peak area measurements also suggested a higher metabolism of the drug by the primary gut model compared to the KCO2 model, and this correlates with the CYP3A4 expression in the models. We have also carried out further calculations to estimate oral bioavailability of a drug and compare them with literature. So results demonstrated that our primary gut liver model demonstrate an improved prediction in oral bioavailability of midazolam in human compared with their KCO2 model. When comparing with animal studies in published literature, our performance in the primary gut liver model is equivalent to the rat and the dog models. So overall, this proof of concept study has provided really promising results and suggested that a primary cells in MPS model can play a huge potential in giving a more accurate estimation on human data. So here are a few take-home points summarizing the potential of MPS technology in contributing to more success in preclinical drug discovery. So first of all, MPS model with incorporation of primary cells has been able to reproduce the physiological conditions of an oral drug dosing in human. And for this reason, the MPS technology demonstrate great potential for more predictive ADME and bioavailability studies. And finally, a hopeful next step would be to combine the gut liver MPS model with computational methods in order to generate some predictive input parameters of PBPK models. And overall, hopefully, with further developments, MPS will be able to offer an in vitro solution to reduce the use of animal testing and improve the overall preclinical drug discovery landscape. So, coming to an end, I would like to give a shout out to Yassine and Audrey, and also our collaborators at Altus Biosystems for all the support. So, if you have any questions, feel free to drop me an email, and I'll be happy to answer them. And yep, that's the end for today. And thank you once again for joining me.